Thank you very much, Jose, and thank you everyone for joining us today. My name is Nick, and as Jose mentioned, I'm a technical application scientist with the software tech support team at Illumina. And today we're going to be talking about enrichment data analysis and specifically the offerings from Illumina. So to begin, we'll go over what this presentation will contain. We're going to do a brief overview of what an enrichment library prep looks like. We're going to move into the different analysis options offered by Illumina, including those on our cloud computing platform, Base-Based Sequence Hub, as well as our local analysis software, Local Run Manager, or LRM. We'll go through what the enrichment analyses are doing in the background, what their workflow is. And then we're going to take a look at some of the key analysis output files, including the all-important variant call files, or VCFs, and some key metrics files produced by the different applications. All right, to start, just taking a quick look at what an enrichment library prep entails. And as it sounds from the name, it does enrich your pool of potential fragments, DNA fragments, to those that you are targeting, things that you're interested in. So this can be used to either analyze more samples in a single run, or analyze single samples more deeply just in the regions that you're interested in. The schematic on the right is showing a biotin streptavidin-based system where we have probes that hybridize to regions of interest. The blue probe is hybridizing to the purple fragment. The streptavidin beads are then linked to the biotin probes, biotinylated probes, to help pull them out of solution, and you're left with just the fragment that you are interested in, and that is the enrichment process in a nutshell. All right, so we're going to go through the different analysis options from Illumina. So we have two ways we can do enrichment analyses. We can do them on our cloud platform, Base-Based, or locally using Local Run Manager. There are different applications within the Base-Based Sequence Hub ecosystem, including the Illumina Enrichment app, the DRAGEN Enrichment app, which has both germline and somatic variant calling, as well as the BWA Enrichment app. And we'll go through in detail what these can do later. And then we also have the option to run analysis locally, either on an instrument if it's supported, or off instrument using Local Run Manager and the DNA enrichment analysis module. All right, so to get into the workflow, what's actually happening when you run an enrichment analysis? These are the main steps. We're going to go into detail on each of them. So we're just going to go from demultiplexing your run data and generating FASTQ files, trimming adapters, aligning to a reference, and then variant calling. Okay, so the first step is demultiplexing the data. So in a typical run, your libraries will be pooled with other libraries, other samples. They'll all be multiplexed on the flow cell. So we have to deconvolute that data. The way that is done is informatically using the index sequences that are part of the library preparation. These are known going into the analysis. If you tell the software which sample has which indexes, it's then able to identify which reads belong to which samples, and that is the demultiplexing process that takes place during FASTQ generation. And once we have our FASTQ files, we need to trim adapters. So adapter trimming is important for a couple of reasons that we'll go into. This can be performed either when you're generating FASTQ files, specifically with our BCL to FASTQ conversion pipelines. There are options to trim adapters during FASTQ generation. It can also be performed as a step after FASTQ generation. There are multiple tools freely available There's also one on base called FASTQ Toolkit, and this you can point to your FASTQ files and give it a sequence and it will trim adapters. There's also the option for some library prep kits to trim within the base enrichment and BWA enrichment apps, but this is limited to certain Illumina kits. So if the kit that was used is not one of these, you would need to trim adapters before running the analysis. Next, we move into alignment. So this is where the reads that have been adapter trimmed are aligned and mapped to the reference genome. This results in an alignment file. Typically, the output is a BAM file. And for all of our pipelines, the BAM file is the main alignment output. We've got a screenshot here of what this file looks like when loaded into the Integrative Genomics Viewer, or IGV. And basically what we're seeing here, the little gray boxes are all the reads piling up over the exons of the EGFR gene shown in blue boxes down below. Everything between those blue boxes are the introns, and this makes sense because this particular panel was, among other things, targeting the exons of the EGFR gene. And at the very top track, we see the mountains, the gray mountains, and those are just giving us a summary of our read pileups over those regions. And this is a typical output we would expect for an enrichment because the probes specifically pulled down the exon regions for sequencing. And I'll say here, and we'll talk about it again, alignment is the reason that we adapter trim is because alignment is trying to match your sequences to the reference genome, and in order to do so effectively, we need to ensure we've removed all synthetic sequences introduced during library preparation. After we've done alignment, we move into variant calling. And here I'm going to break down the different applications that Illumina has, what they can and can't do. So you have a synopsis here. Depending on what kind of analysis you want to run, you'll be able to tell which application is best for your needs. So we have our base enrichment app. This can perform small germline and somatic variants, as well as small indel calling, insertion, deletion. It can perform copy number variant analysis and structural variants, and we'll get into some of the differences there. Next, we have our local run manager enrichment module. So this is for local analysis. This can do either germline or somatic small variant calling and small indel calling. The base BWA enrichment app will only do small germline variant calling, so single nucleotide variants and small indels. It does not have a somatic caller. And lastly, our base-based DRAGEN enrichment application can perform small germline and somatic variant calling, as well as copy number variant analysis and structural variant analysis. And I'll note here that with the addition of version 3.6.3 of the DRAGEN enrichment application, there is now the option to enter in command line options that one might use with a DRAGEN on-site server. It's use at your own risk, but you are able to enter in command line options into one of the fields on the app launch page to further customize your analysis. Okay, so we're going to take a look at the some of the input files we need for the different applications. So for the base enrichment and BWA enrichment apps, as well as the enrichment module for local run manager, in addition to our FASTQ files as input, we also have to provide what's called a manifest. And what we have here is a picture of an Illumina manifest. This is an Illumina specific file format, and we'll break down the different columns that we're seeing here. So in the first column, we have our target region name, and this is just the name given to that region. And the region is defined first in the chromosome column. So here we have the chromosome or contig number, here just showing chromosome 1. And then the next two columns are the coordinates, the start and stop coordinates, or start and end coordinates for that region. So the chromosome start and end columns define the region, and then the name column gives that region a name. And because this is an Illumina specific file, if you're using an Illumina enrichment kit, the file will be provided either with the kit, or usually you can just download it from our support site. If you are running a custom or third-party kit, we do have an option to generate one of these if you have a bed file, because most other third-party kits will come with a bed file defining those target regions. And speaking of bed files, so this is a common file format in the industry, and it's similar to the manifest we just saw with some key differences. And we're bringing it up here because the DRAGEN Enrichment app actually uses a bed file directly, so we don't have manifests for these. They're defined, the regions are defined in bed format. And again, we'll just go through the columns here. So in the first column this time, we have our chromosome or contig. So here we have some from chromosome M, or the mitochondrial chromosome, and then chromosome 1. Then columns 2 and 3, we have our start and stop coordinates. And then in the fourth column here, we have our target region name. The first three columns are required for a bed format file. The fourth column here can be added, and here it will help distinguish which region is which just by looking at the name. And again, this is the type of file format that the DRAGEN Enrichment app will take. Okay, so we're going to go through the different types of analyses within each of these, starting with small variant calling. So all of our enrichment analysis options can perform small variant calling. That's SNVs, or single nucleotide variants, and indels, or small insertions and deletions. With the base enrichment app and local run manager enrichment module, any variants outside of the defined target regions will be labeled off target in the VCF. So what that means is anywhere that reads a line to the reference genome, the enrichment module and app will attempt to find variants. If they do find a variant that is called, but it is outside of those target regions, it will still print it in the VCF. It will just label it as off target, and we'll show an example of that in a few slides. With the DRAGEN Enrichment app, variants outside of the target region are not included in the VCF. So it restricts the variants that are output to just those within the defined target regions. There is, however, an option to pad those regions or add bases on either side, so upstream and downstream, to tell the DRAGEN application to look for variants in those padded regions flanking the target region. And by default, this is set to 0, but you can increase it. So you can add, say, 150 bases up and downstream, and the variant caller will then report on any variants found within those flanking regions, padding the target region. Next, we have copy number variant analysis, or CNV calling. So with our base enrichment app, it can perform CNV calling. It uses Illumina's Canvas caller, that's the name of the algorithm. And this does require at least one control data set. So you have a control data set with copy numbers that you know or what would be considered normal. And you can use that to set a baseline that Canvas can then use to determine if there are changes in copy number in your experimental sample. Similar to that, in the DRAGEN Enrichment app, the CNV caller is the DRAGEN CNV caller. This does require pre-building your control data set. And what I mean by that is you can feed in your quote-unquote normal samples into the DRAGEN CNV baseline builder, which is a separate application. And then the files produced by that can be fed into the DRAGEN Enrichment app, and those will be used to set the baseline. I say here that there's a maximum of 10 controls. That is a limitation of the input for the DRAGEN Enrichment app. If you want to build many more baselines using the baseline builder app, you can do that. It's just when you go to launch the application, we've limited it to 10 controls, which should be sufficient. We would highly recommend using more than one, however, just to make sure that there's no issues and that you're getting a good baseline. And similar to the canvas color, it's going to use that baseline to determine if there are copy number changes. And then lastly, we have our structural variant calling. So both the base-based enrichment app and the DRAGEN germline pipeline use Illumina's MANTA structural variant caller. This is to find larger insertions and deletions as compared to the small indels done by the small variant caller, as well as inversions and tandem duplications. Okay, so those are the different types of variant analyses going on in the background. And now we're going to take a look at some example outputs from these different analysis applications. Just to summarize again, we have our main output files that's typically starting with your BAM file, so your alignment file that contains all those reviewed pileups in reference to the genome that it was aligned to. We have our VCFs and genome VCFs, if that is an option that's selected. We'll talk more about those in a moment. Then we have our CNV VCFs, so a separate file that will tell us if we see any copy number variations, and as well as the structural variant VCF, again, telling us if there's any structural variant changes. And then some of the other useful output files we'll go into. These are things like the coverage and summary and sample metrics files produced by the respective applications. And we'll go into those a little bit and how they can help with troubleshooting. All right, so here we have a typical VCF file. And we're going to go through each column and break down what it's showing us. So we first start with the position of the variant, and this is defined in two columns. First, the chromosome column, telling us what chromosome it's on, and then the position is the coordinate, so the single base position where we're finding that variant. We then have the reference and alternate, or ref and alt columns, and what this is showing us is the ref column tells us what the base call is in the reference, so what that base is in the consensus reference sequence. And then the alt column is telling us for our sample what the alternate base or allele was found to be. We then have our filter column, and this can tell us a lot about the variant. It can tell us if it passed all variant filtering steps, which would be denoted with a pass, which you can see in the top variant. If for any reason a variant was determined to not pass any of the filtering steps, the reason will be encoded in the string that's printed in the filter column, for example, off target or low GQX. As I mentioned before, if the variant is identified outside of the target region defined in the manifest or bed file, it will be printed as off target. So it might be something that is the true variant, it's just not included within the defined target regions. And then something like low GQX would be low genotype quality. So that is another filtering step that the variants need to pass. And then lastly, we have our format and sample columns, and we'll go through some of the keys in here. Basically, what you have in the format column is a list of metrics that are encoded with these acronyms. And then in the sample column, those metrics are defined or given values. For example, the GT is the genotype, and this is just telling us what the genotype was determined to be at this position. So for example, again, if we take a look at that top variant, the genotype is reported as 1/1. So a 0 is a reference call and a 1 is an alternate call. And if it's 1/1, that means it's homozygous for that alternate allele. In this case, the reference is an A, and this sample was found to have Gs at both alleles. And that's why we get the 1/1. Then we have our depth, denoted as DP. And again, we'll take a look at that top variant. We see that the total depth, total reads used to call this variant was 124. And next, we will move to the allele depth, or AD. And again, I'm going to go up to that top variant. What we're seeing here, instead of a single number, we have two numbers separated by a comma. And what that's showing us is the number of reads, number of that total depth that were found to be either the reference or the alternate. And not surprisingly here, we see 0,124, which is telling us that no reads were found to have the reference sequence at this position, and all 124 were found to have the alternate allele. Okay. So in addition to the VCS that are output by our base-based enrichment app, on the reports page, there's a cool function called the variance table. And basically what this is showing us are our variants as we would see them in our VCF, but it's an interactive table. So if you look at the white boxes at the top of every column, those are actually text fields you can enter text into. And so if you want to say, limit things to just chromosome 2, in that very first one on the top left, you would type in CHR2, and it would live update to filter just those variants found in that chromosome. Similarly, if you were looking for any variants found in a particular gene, you can enter that gene symbol in the gene column, and it will filter for variants that were found just within that gene. So it's a nice way to quickly look at things, narrow it down if you want to just take a quick look at your called variants. And I will note here that the variants printed in this table are only those that passed filtering steps. So we saw in the last slide that some of the variants had the word pass in all capitals. Those are the ones that will make it into this variant table. If for any reason they're filtered out, they may still be included in the VCF. You just need to go look at that file. Okay. So we're going to take a look at the small variant calling output, particularly the genome VCF files. And we're going to discuss a little bit about what it can do for us, the differences between that and the VCF file. Basically, what the genome VCF is showing us is information on a site whether or not a variant was found. So in the VCF, it's only reporting those where variants were called. Then it tells us whether or not they passed, but the variant had to be called. Whereas in the genome VCF, it doesn't have to call a variant. It still reports information found at that particular region. And because of this, genome VCF files can get pretty large pretty quickly. So depending on your sample depth and the analysis type, these can range in size. They can get into the many gigabyte size of files. So just something to keep in mind. And we'll take a look at what the genome VCF format looks like. So the base-based enrichment and local run manager enrichment applications will create both VCS and genome VCFs for every sample. That's how the program is designed, and that's the output that you'll get. With the base-based DRAGEN enrichment, you have some more choices. It will generate a VCF and genome VCF for germline. You also have the option to add or to have an output in base pair resolution or BP resolution. This is typically used for troubleshooting and debugging. Basically, it's going to give you information at every base pair, so the resolution is every single base pair, rather than a banded VCF that includes information for larger swaths of the genome. Again, these will get very large very quickly, so typically used for troubleshooting or debugging. And then lastly, for the somatic, if your variant calling mode is somatic, it will only generate a VCF. We don't have options for genome VCF or base pair resolution there. Okay, so this is what a genome VCF looks like. And you can see it's pretty similar to the VCF we saw before. There are some differences here. I'll note in the ref and alt column, we see that for most of the alt, it's just denoted with a dot or a period. And this is just showing us that it didn't find anything different from the reference. We do see at the 6th position down, there's a T in the alt column. So what this is showcasing is that the genome VCF will include all of your variants that were called. It will just include a lot more information. So essentially, the VCF is a subset of the genome VCF. Similar to our VCF, we have our format and sample columns. And again, we have things like depth or DP. And we're going to take the use case of the very last item here. So we have a position here where no variant was called. The reference is an A. In the alt column, it's a dot. The depth is showing us as 282, which could be sufficient. So say we're expecting a variant in this position, but we get this output by the applications. This might be something we can look at to determine if we have enough depth to call the variant at that position. This could be that either there is no variant here, and it is a true reference call, or it could be that it's a very low frequency variant and we just need more depth. We need to sequence deeper at that position in order to recover that variant. With the genome VCF, let's take a look at the CNV and structural variant output from the base-based enrichment app. So we have here, and you can see it looks similar to the VCF format that we've seen before. We just have some differences, particularly in that ID column. So again, we have our chromosome and position. We have our ref and alt, and these look a little bit different as well. And then we similarly have our filter column. So if we take a look at this variant here, we can see that Canvas in the ID is calling it as a gain. And again, if you'll remember, Canvas was our CNV caller for the base-based enrichment app. The reference or the alt is being shown as CNV. So rather than having a base and then a different base, because it's a larger region of multiple bases, it's reported here as a CNV as the alt. And then again, in our filter, we have some things coded here, and these may not mean much, Q4, CLT 10KB. However, either in the documentation or in the VCF header, so in the same file at the top of the file, these should all be defined. So anything that you find in the filter column, you should be able to find more information about it in your VCF header, as well as the documentation for that application. And with the base-based enrichment app, it will actually combine structural variant and CNV calls into a single VCF file. So here we have MANTA, which again is the structural variant caller from Illumina. And this is denoting an inversion, or INV. And this one actually passed in the filter column. We see that it passed all the filtering steps. It has a pretty high quality. So this is where you can expect to see your structural variant calls as well. And I don't have an image of it here, with DRAGEN, you will get two separate files, one for the CNV calls and one for structural variant calls. Okay. Let's going through our VCS, GVCS, as well as our CNV and structural variant VCS. We're now going to take a look at some of the other useful outputs from the different applications and with an interest in how they can help us with troubleshooting. So one that is pretty useful from base-based enrichment and local run manager is the coverage CSV file. And this is showing us our target region, so the chromosome start and stop, as well as the region ID. And then it gives us the mean or average coverage at that position. So this is telling us the average amount of coverage within the defined target region. So if you have a variant that you're expecting, you're not seeing it, called within the region, you can take a look at this file and maybe you see that you only have, say, 33 as your mean coverage, and that might just not be enough to make that variant call. So this is a nice file to take a look at, especially if you start to need to troubleshoot why your variant calls are not what you're expecting. And some other metric files output by these two applications, the summary CSV files, and these are just snapshots. There's more to each of these files, but we're just giving you an overview of the kinds of things you can find here. So the format is that we have our metric defined in the first column, and then the value for that metric defined immediately to the right. For example, we have here percent aligned reads. This is always a good metric to take a look at. We're getting just above 96% for this sample, which is great because we are expecting to align to this human genome because it was a human sample. So the fact that the majority of our reads are aligning is a great first step to understanding how our sample is doing. Another type of metric we can expect are things like the target coverage at a certain depth. This is showing us that just under 92% of our targets are being covered at least 20x. So it kind of breaks it down from 1x all the way up to 50x. So you can take a look at how your targets are being covered overall throughout the entire panel. And then similar to those outputs, this is an example of the DRAGEN enrichment output. And here we have, again, our metric on the left. And then the two columns immediately to the right give us the values. The first of those two columns is the number, and then the last column there is the percentage. So it gives you the information both ways. And similar to the previous slide, we have, instead of percent aligned, we have our mapped reads. So this gives you the total mapped reads as well as how that breaks down as a percentage of the total. So here we have just under 98% mapping to our reference genome. And we also have similar information to the summary CSV files from the base-based and local run manager apps, here with the DRAGEN output. We've highlighted here that the percent of the target regions with a certain amount of coverage. So this is just showing us that about 45% of our targets had a coverage of 100x or more. This is also nice information just to see how your prep is performing. Okay, so we're going to talk about some key metrics here. So we'll go into these a little bit more detail. We're going to look at alignment metrics. We've already talked a little bit about the percent reads aligned. We're going to talk about on-target alignment, coverage uniformity, and duplicate read. So with the base-based enrichment and local run manager enrichment modules, the report will output something like this, where we will get our percent aligned reads for that sample, as well as the read enrichment. And so the read enrichment is the percentage of the aligned reads that aligned within the target regions. And if we look at the padded read enrichment there at all the way to the right, that's actually showing us the enrichment if we extend the target regions. A typical padding is 150 base pairs up and downstream. So this jumps from 82% to 88, which we'd expect for an enrichment because a lot of times those flanking regions just outside of the target regions will also get pulled down during the library prep. Okay, and for the DRAGEN enrichment, this is an example of the kind of output we would see. Again, we have our mapped reads here. And then here we have, instead of read enrichment, we have something called capture specificity based on the target region. You can read that as percent enrichment. Here it's given as a decimal. We can consider this 78% read enrichment. Some of the things we can use these metrics for in troubleshooting is if, say, we have a low percentage of aligned reads overall. One of our first questions will be whether or not adapters were trimmed. As I mentioned at the beginning, if synthetic sequences left over from the library prep are still included in the reads, the aligners will often have a hard time aligning that properly with any type of confidence to the reference genome. So that's always the first thing to take a look at, and that's pretty simple to determine. There's several different tools that can tell you whether or not adapters have been trimmed. So say we did trim adapters, we're still getting low percentage of aligned reads. So what we want to understand then in that case is, what do these reads that are not aligning to the genome, what are they aligning to? So with something like that, we can take a look at the BAM file, which will include all the reads, but will flag the ones that don't align. We can take those unaligned reads, and then we can compare them, maybe do a blast search and see what kind of organism they're aligning to. Perhaps that will tell us maybe there was just a sample contamination somewhere along the way. Now say we have a high percentage of reads aligned, but our enrichment or aligned on target is low. One of the first questions we'll ask is if the correct manifest or bed file was used. So if you have good alignment, but your alignment on target is low, it might just be that the regions defined in the manifest file are not those that were actually probed for with the prep kit. And that's usually a pretty simple fix. You basically get the correct regions file or manifest file and rerun the analysis. Say you did use the correct manifest, we would also want to check upstream if the correct oligos or probes were used for enrichment. If there's a lab that's running lots of different panels, it's always possible to accidentally grab the wrong set of oligos and maybe you enriched for something else that was unintentional. And then lastly, if all of those are correct, we got the correct probes, we have the correct manifest, there could be an issue with the enrichment step, perhaps during the pull down or washing steps, things weren't efficiently separated out from the bulk sample. And so we still have a lot of leftover reads not within our target region. Okay, and to help with understanding our coverage, we get some coverage metrics that are output by the base space and local run manager applications. We have our mean target coverage depth. So this is taking all of our regions and giving us the average coverage across all of those. So this can be high, but still might not solve all the problems. And that is where the uniformity of coverage metric comes in. So basically together, these will tell us if A, we have good coverage across all our regions, good average coverage, and B, in the uniformity of coverage, it will tell us whether that all of our targets are covered at roughly the same amount. So rather than, say, having one region that has thousands of reads aligning to it and it's offsetting the average, the uniformity of coverage will give us an idea if all of the regions are getting roughly equivalent amounts of coverage. And with the DRAGEN output, we have, again, we're taking a look here at the percent of the target region that have a certain amount of coverage. This, again, can give you an idea of that uniformity of coverage. So if you see that 90% of your targets are only covered between 10 and 20x, that's going to give you an idea of maybe that we just have low overall input, things like that. And it will also give us the average sequence coverage over all our target regions. So if we do, say, have a low coverage of our targets, as I just mentioned, it could be that we have maybe a low number of input reads. That could be because of low sequencing output, maybe poor demultiplexing. It could be poor alignment. And for this, we would want to take a look at, again, just see if maybe we just have a low amount of reads coming off the sequencer. If too many samples were pooled onto a flow cell, perhaps they all just have low output. So we might need to address some things when loading. For the coverage uniformity, if we say have regions with really low coverage and others with decent or high coverage, we might want to take a look at the input samples and input quality of those samples. If they maybe are degraded, maybe some of those regions that we're targeting have been lost just due to sample quality. All right, now we're going to take a look at duplicate reads. So this is a metric that is defined as read pairs with identical start and stop alignment coordinates. These can be introduced during PCR steps of the library preparation because they were essentially making copies of the templates we have there. And both of these applications will identify duplicate percent reads. So here we have an example from the base space and local run manager enrichment app. And we have about 12.5% of our reads were flagged as duplicates. And an example from DRAGEN, we have just under 18% were found to be duplicates. Now, this is not to say this is an issue, just knowing these numbers here. What might be concerning is, say, you run the same panel multiple times, and then you start to see 18% enrichment, or sorry, 18% duplicate one day, and then on another run, you're seeing 60%. That would certainly indicate an issue that we'd want to address. So if I have a high percent duplicate in my sample, what are some of the things we can take a look at? Again, we often come back to the input material. If you just have a low amount of input going in, those will tend to be copied more during those PCR steps because there's less template available. So just the chance of introducing PCR duplicates is a little bit higher with those lower input or maybe lower input quality samples. We want to understand if enough of the library was added to the enrichment step. So if just not enough library was added during enrichment, perhaps it's not a good representation of all of the possible fragments that the panel is probing for. And then lastly, we always want to check maybe there were extra PCR cycles added during library prep. In labs, machines are getting used by multiple people. Maybe a protocol got changed and some extra PCR cycles were added. So it's always something to check out because this can certainly introduce these types of artifacts. All right, and in summary, these are the different applications that we went through today. The base space offerings, enrichment, BWA enrichment, and DRAGEN enrichment, as well as the local analysis option with local run manager and the DNA enrichment module. We went through all the different variant calling that these can do, and the importance for running certain applications for the output that you desire. We also have included at the end of this slide deck some links to information and resources for you that you can take a look at. And if you do have any questions, you can feel free to ask us here. You can always call Illumina technical support, and we'll be happy to help you out. And with that, I'm happy to take any questions.